Hello, welcome back. Did you miss viruses? You know, they didn't miss you. So now we're going to resume talking about how viruses cause disease. Today we're going to talk about adaptive immunity. You know, this is a very superficial, cursory look at this because it's not the focus of this course, but we have to know some of these things to understand not only general things about pathogenesis, but we're going to start talking about specific viruses in a bit, and you really need to know something about the immune response. So just to remind you where we are, we've talked about a variety of host defenses so far. Last time we talked about intrinsic defenses, which are always present and include things like apoptosis, autophagy, RNA silencing, and antiviral proteins like tetherins and apobex. We talked about the innate immune system, which can sense a foreign protein or nucleic acid and respond by producing cytokines, including interferons, which then induce antiviral proteins. And we said that the innate immune system is induced by infection, but typically doesn't have memory. And then today we were going to talk about the adaptive system. And the name tells you what it's about. It's tailored to the pathogen. So it is specific for a given virus or bacterium, whatever you're infected with. And it has memory. And of course, that means that the next time you are infected with the same virus, this immune system will remember and respond quickly and with great vigor to prevent infection. So we'll talk about that today. And just to remind you how we get to the adaptive system. We have an epithelial surface here on the left. Virus infects the cells, respiratory epithelia, gut epithelia, any part of your body that initially gets infected. Virus enters the cells, replicates. The cells sense the presence of virus. Remember, there are toll-like receptors on the cell surface in the endosomes. There are cytosolic receptors that sense the virus is present, and the cells then make cytokines in response, the red dots here. And those cytokines, many different cytokines, including interferons, which of course will then induce an antiviral state in the neighboring cells. The cytokines that are produced will also attract immature dendritic cells to the site of infection. Remember, these are cells that are patrolling all different areas of your body, and they will be attracted to infected cells by the cytokines. They have cytokine receptors on their surface. They have toll-like receptors. They have innate sensors of all sorts. They can produce interferon on their own, and they do. They can make interferon to try and eliminate the infection. But they will also pick up packets of proteins and nucleic acid released from the dying cells and determine if they are foreign or not. And if this early event, this innate defense, cannot control the infection, then the dendritic cell will go to the lymph node and activate the adaptive response. And that's what we're going to talk about today. So these dendritic cells, we talked about this last time, when they pick up signals from the infected cells, they become matured. They look like the typical dendritic cells. They go from the site of infection through the lymph capillaries. They reach a lymph node, and there they can encounter both B and T cells, which will lead to the production of antibodies, or T cells, which are the mediators of the adaptive response. Now let's do some terminology first to make sure you understand the words that I tell you. And there are two words that we throw around. Let's make sure we understand what leukocytes and lymphocytes are. Leukocyte is a general term for white blood cells. And white blood cells include all of these shown here: lymphocytes, neutrophils, eosinophils, and macrophages. And these are drawings of what they look like. They can be distinguished under a light microscope. So that's a leukocyte. A lymphocyte is a subset of these leukocytes and includes T cells, B cells, NK, natural killer cells, which we talked about last time. And these are distinct because they have variable antigen-detecting cell surface receptors, and we'll talk about those today. That's what distinguishes the lymphocytes from all of the other white blood cells. All these cells and more arise from a common progenitor, a multipotential or multipotent hematopoietic stem cell, which is self-renewing. It's remarkable. From this one stem cell, all these cells arise, including the ones that I've just been talking about. And early in differentiation, these stem cells can go to the myeloid lineage. They can differentiate into a common myeloid progenitor, which gives rise to the myeloid lineage, which includes erythrocytes, neutrophils, eosinophils, macrophages, basophils. And then on the other side, they can go towards the common lymphoid progenitor, lymphocytes, including NK cells, T cells, and B cells. So those are the lymphocytes on the right. And some of these we'll be talking about today. Now remember, again, this is from last time, but I want to repeat it, how a dendritic cell becomes activated. So on the left, we have a immature dendritic cell. These are the sorts patrolling all of our tissues, and they have, as I said, receptors for foreign proteins and nucleic acids like toll-like receptors on the cell surface and within the endosomes. They have MHC class II proteins, which are going to be an important part of what we talk about today. They can take up foreign material by endocytosis. They can tell if it's foreign or not. They can respond by making cytokines. But they also can process foreign protein and display it on MHC class II molecules. We'll look at that in a moment in detail. They would take up proteins from a dead cell, for example, and they would be endocytosed and mounted into MHC class II. And as these dendritic cells are picking up the signals from cytokines, they're becoming activated, and they become the cell on the right. And as I said earlier, they make interferon. Again, the interferon at the site of infection to try and clear the infection. They become activated. They display viral peptides on the surface. This is the indication that we have a viral peptide, these little orange boxes being displayed in MHC, major histocompatibility class II receptors on the cell surface. And these dendritic cells, once they're activated, they will go into the lymph node, a local lymph node. So if you have a respiratory infection, they will go to the lymph nodes nearest to that, and they will interact with B and T cells. And here we have shown the dendritic cell interacting with a specific kind of T cell that eventually is going to be a helper T cell. And the way these interact is via a series of receptors and ligands on both the dendritic cell and the T cell, including the T cell receptor, CD28 and others. But what you can see is happening here, the dendritic cell is presenting the viral peptide, or the peptide in orange, we don't know if it's viral at this point, to this T cell. The T cell is binding that peptide with the T cell receptor, and a lot of other receptor interactions that are going on here. And the T cell will determine whether it's foreign or not by a process we don't have time to go into, which is fascinating. And if it's foreign, the T cell will become activated and differentiate into what it's destined to be. In this case, it's a helper T cell. So this process is guided by the peptide produced or displayed by the dendritic cell, and also by cytokines that are produced by the dendritic cell as well. They help to instruct the T cell and activate it if necessary. So if it has a foreign peptide, if it recognizes that this is a foreign peptide, and that means not self, then the T cell will become activated. All the T cells that recognize self should in theory be eliminated during development in the thymus. All the self-recognizing T cells are killed by apoptosis. So what is left should be just ones that recognize foreign. Of course, due to a variety of mutations and diseases of various sorts, some of us retain self-recognizing T cells, and that's a problem. So here we're showing the dendritic cell interacting with a T cell that's going to become a helper T cell. It could interact with a precursor of CD8 positive T cells. So the helpers are marked by CD4 on their cell surface, and the CD8 positive T cells can also interact. They will end up being cytotoxic T cells, as you'll see in a moment. And the dendritic cell can also instruct a B cell. It can present a peptide, and if the B cell recognizes it as foreign, it will become activated and lead to the adaptive response. So that is the interaction between the innate and the adaptive response. It's very important, and of course, the dendritic cell being here is part of this whole inflammatory process by which infected cells make cytokines. They lead to those 4 signs of inflammation, and that's when you get a good adaptive response. Now let's talk about the presentation of antigen by those dendritic cells. Remember, the dendritic cells are taking up pieces of dead and dying cells that are killed by virus infection. It processes the peptides and presents them on MHC class 2. This is called the exogenous antigen presentation pathway. The name is very straightforward. It simply means the cells are taking up exogenous protein floating around in the extracellular milieu, and then processing it in the cell. And that's going to be in contrast to the endogenous pathway, which we'll see in a moment, where a virus infects the cell and the proteins are made inside the cell, and those are then put on cell surface MHC. So you can imagine that this might be a dendritic cell or a macrophage, an antigen-presenting cell, which is phagocytic. It's taken up protein, again, released from dead and dying infected cells. So you see antigen-presenting cell here. The protein is taken up by the endocytic pathway, the same pathway that we talked about that takes up some viruses. Proteins are brought into this pathway. The endosomes become late endosomes. The pH drops, and the protein eventually gets chopped up by proteases in the endosome. Meanwhile, these cells also make the MHC2 protein. And because this MHC2 is destined for the plasma membranes, that blue protein there on the right with the peptide in it, it's made in the endoplasmic reticulum initially. It is shipped through the Golgi, and it intersects with the endosome and picks up peptides that have been processed and brought in from the extracellular milieu. And so you can see this particular MHC molecule is picking up an orange peptide. It's then going to be shunted to the surface in a vesicle, and that MHC will display the peptide on the cell surface. So that is the exogenous antigen presentation pathway, whereby an APC, a dendritic cell or a macrophage, will take in protein from an area of infected cells, process it, display it in MHC2, and of course the next step is going to be, it's going to go in the lymph node and present that to T cells and B cells. Now, just like with innate and intrinsic defenses, viruses interfere, or they encode for gene products that interfere with MHC2 and MHC1, as you will see, processing and presentation. In particular, a herpes virus, human cytomegalovirus, interferes with transcription of MHC class 2, so that prevents the display of peptides, viral peptides, on the cell surface, and of course that's going to inhibit an adaptive response. And human cytomegalovirus, another virus that all of you eventually will have, causes lifelong persistent infections. It's not cleared when you're initially infected because of mechanisms like this. It evades immune clearance. This pathway, the MHC2 pathway, is restricted to only a few cell types in the body, including antigen-presenting cells. And there are not a lot of viral antagonists of this pathway. This is one of the few here. And as you'll see in contrast, the MHC1 pathway is far more antagonized. So that brings us to the lymph system, which we've encountered before in this course, where in terms of viruses spreading from the initial infected area, getting into the lymph system, and then getting into the blood. Now we're going to talk about it in terms of adaptive responses. So when the dendritic cells enter the lymph node with a peptide displayed in MHC2, again, they interact with B cells and T cells, and here's a lymph node in the middle, and it has a number of different areas which are devoted to the development of B cells, the primary lymphoid follicle, the paracortical areas, mostly T cells, and so forth. And that's where these dendritic cells will go in to interact with these two cell types. You have an extensive lymph system in you, as you can see here on the right. It's part of it drawn there, consisting of lymphatic vessels, capillaries, and lymph nodes. You have lymph nodes scattered throughout you. And the reason is that they need to be near areas where there's immune surveillance for infection, so that the dendritic cells don't have to travel very far to get to them. Now, in your circulation at any given time, somewhere between 1 in 10,000 or 1 in 100,000 T or B cells are specific for any given antigen, say from a virus. So there's not a lot, and it's circulating through you at any given time. They're in your lymph nodes. And so when the dendritic cells come into the lymph node, if they are bearing a foreign peptide, they will activate either B or T cells, and they start proliferating. So that within 1 or 2 weeks, there's between 1,000 and 50,000-fold amplification of the number of cells. You go from 1 in 10,000 to 50,000 times more. It's a huge amplification. As a result, the lymph nodes swell. If you've ever had a sore throat and you feel underneath your chin, you feel swollen lymph nodes. There are 2 prominent ones right under here. You can tell if you got a little local inflammation because they're swollen. That's called lymphadenopathy. And it's a key finding in many infections. If you get a common cold, you're going to get swollen lymph nodes underneath. Of course, there are many other parts of your body that the lymph nodes will swell, but you can't feel them as readily as you can under your chin here. If you get a GI infection, the local lymph nodes will swell, but you can't tell that either. Lymphadenopathy was one of the early signs is still one of the early signs of HIV infection, as we'll see later. So that's what happens when the DCs bring the antigen in and activate T or B cells. The cells proliferate, got to make more of them, of course, and that's why the lymph node swells. Lymphadenopathy. All right, our first question is, what is a property of innate instruction of adaptive immunity? A, presentation of viral peptides on MHCI to CD8 T cells. B, endocytosis of viral proteins. C, activation of dendritic cells by cytokines. D, sensing by toll-like receptors, or all of the above. Answer is all of the above. So most of you got that, but many of you picked presentation of viral peptides. That's certainly true, but they're all true, so that's why you have to pick all of the above. Remember, the peptides are presented after the proteins are endocytosed. The DCs have to be activated by cytokines before they will present, and TLRs are all involved in the sensing as well. So it's the whole thing, it's all of them. So here's an overview of all of the effectors of the adaptive response that we're going to talk about today. Let me give you a kind of a 10,000-foot view of what's going on here. So we've talked about a variety of antigens being presented by dendritic cells. It's a dendritic cell, the orange cell there on the upper right next to the mouse. And so here, the animal is infected, in our case viruses, but can be any antigen coming into the animal. The dendritic cell will process the proteins, bring them into the lymph node, and then turn on the T cell arm or the B cell arm. So let's take them separately. Let's go on the left first. In the lymph node, the dendritic cell can present the viral peptide to B cells. And that will eventually lead, if the peptide is foreign, to the production of antibodies. Those are the little red Y molecules, which are initially bound to the membrane of the B cell, but once the B cells become activated, they can differentiate into what we call plasma cells, which make many, many antibodies, huge numbers of antibodies which are secreted, and those antibodies can interact with viruses. And we'll talk about the consequence of that in a minute. The process depends on a number of things happening. First of all, the antigen has to actually bind to antibodies that are bound to the surface of the B cell, shown here on the left. And so that can be presented by a dendritic cell. That binding and cross-linking of the antibodies on the cell surface. Remember, this is one B cell that just happens to have a specificity on its surface that recognizes the foreign peptide. That leads to transcription in the nucleus of a variety of genes that are then devoted to making antibodies. And as you know, if you've taken immunology, the activation of B cells involves a recombination of the genes encoding antibodies, eventually somatic mutation to make very high affinity antibodies and so forth. But we can't talk about that today. Besides the antigen binding to the immunoglobulin on the cell surface, so I'll use IG or antibody interchangeably, the production of antibodies requires help from T helper cells. So here is a CD4 positive T cell, which is presenting the peptide to the B cell at the same time. This leads to the production of cytokines, helper cytokines that are important for the maturation of the B cell. But in the end, the real key here is that these B cells respond to the foreign peptides by the production of antibodies, the little red Ys, and those are going to be important for dealing with the infection. The other part of the adaptive response is the cellular response, and that's shown here on the right. Again, the dendritic cell is coming in the lymphocyte and activating T cells. Before that happens, we have in the bone marrow naive T cells. This is where they initially develop from that hematopoietic precursor stem cell that I showed you earlier. They migrate to the thymus, where they undergo additional maturation to become CD4 and CD8 precursor cells, and then they migrate to the lymph node where the development is completed, and there they are CD4 positive, and these are T helper cells. We also refer to them as T little h, and they mainly produce cytokines that are involved in other immune responses. For example, we talk about Th1 cytokines, which help the maturation of CD8 positive T cells to cytotoxic T cells, which are going to kill infected cells. So the CD8 T cell, or the CTL, cytotoxic T lymphocyte, its job is to kill virus-infected cells, and we'll talk about that in a bit. And so it requires, to develop, those CD8 cells require Th1 cytokines produced by helper T cells. And then there's the Th2 cytokines, which are involved in the maturation of the antibody response, as you can see here. And we also saw a Th2 response on the left side here, producing cytokines that helps maturation of the antibody response. So two kinds of lymphocytes, one mainly producing cytokines that help with CD8 cell development and antibody development, and then the CD8 positive T cell, or CTL, which is involved in killing virus-infected cells. And so the two effectors that we'll mainly be concerned with today are the antibodies made by B cells and the cytotoxic T lymphocytes. We're going to talk about how they prevent or resolve virus infections. But the whole process depends on these helper cells. And in fact, as you'll see, for some viruses, antibodies are more important than CTLs, and for other viruses, CTLs are more important. And these helper cells are key in determining what kind of response you get. Because if a virus is best neutralized with an antibody response, then the T helper response is likely to be a Th2 response, because it's been adapted to the virus itself. And contrarily, if the virus requires CTLs to be cleared, you're mainly going to have a Th1 CD4 response. And we'll talk about that in a bit. Let's look at antibodies, or immunoglobulins. In us, the antibodies have this Y-looking appearance. They're made up of 4 separate polypeptide chains, 2 heavy and 2 light chains. And they are attached or associated with each other in a fashion that is approximated by a Y. And these molecules have hypervariable regions, which are the parts that are interacting with antigen. And the rest of the molecule has a structural formation or function. At the top, the Y part is called the FAB, and this is the part that binds antigen. The bottom part is called the FC region, and this region binds to receptors on host cells called FC receptors, and that can mediate other functions. In addition, you can see here in the FC region, there's a complement binding region, because antibodies are used in the complement system to help clear pathogens. Now, when you are immunized with an antigen, this is the response that you get, shown here on the right. So on this graph, we're looking at the serum antibody titer. So you take blood out of an infected animal, for example, and you separate out the serum from the cells, and then you ask, what are the antibodies in it? You measure it in some way. And this is weeks after inoculation. So if this were an experiment, we give the animal antigen A, and then we measure the primary antibody response in red. And so you can see after about 2 weeks or so, there is an antibody response. The first one, it's called the primary response. And then after time, the titer of antibodies declines. So it never remains high for a long period of time, but there is some titer there. And then if you would, in this case, give the animal another injection of the same antigen, you would see a very rapid and more robust response. Because if you compare the time it took to get the primary response, the secondary response is much quicker and it's higher titer. And that's the memory response, because all these B cells have already been selected after the first antigen. They have been amplified. The immunoglobulin genes have rearranged to make high affinity antibodies specific for the antigen. And then all that information is stored as a memory B cell, which remains in us. And then when the antigen is encountered again, we can have a rapid response and a much higher titer. And the idea is that that would prevent an infection, whereas the initial time you wouldn't be able to prevent the infection because it takes 2 weeks to get an antibody response. Now, if you mixed antigen A plus a new antigen at that second inoculation at about 7 weeks there, you get our secondary anti-A response, which is quicker and higher because of memory, and you would get a primary anti-B response. So they are separate. The antigen determines whether you're going to get a primary or a secondary response. Again, we're measuring in this experiment antibodies in serum, but they are throughout us. They can diffuse through various tissues. They're present at mucosal surfaces as well. So here's an experiment where a host animal was inoculated with poliovirus, and then at different days after immunization, we are measuring the antibody titer in the serum against poliovirus. So you would, again, take blood out, separate out the serum from the cells, and then you would mix it with virus and ask, is there antibody present in the serum that is going to bind poliovirus, reflecting its specificity for the virus? So here on this graph, we are showing 3 different kinds or classes of antibody, IgM, IgG, and IgA. And these have different roles in our adaptive response. As you can see, the first antibody to be made is IgM, which goes up rapidly, peaks at about 20 days, and then declines. And IgM consists of 5 dimeric antibody molecules joined together by their Fc regions. The IgM is often used as a diagnostic to see if you've been recently infected, because it goes down in a couple of weeks, and is replaced by IgG, which is the green line here, which you see rises a little slower and goes to higher peaks. So if you measure someone's poliovirus antibody and you see only IgM, that's a recent infection. Whereas if you find only IgG, the infection is older. It's a great way to determine or distinguish between the two. The reason IgM has this pentameric structure is because the antibodies made early on are not of very high affinity. They still have to, the B cells that make them still have to go through maturation, somatic mutation, and cycles of selection for higher and higher affinity in the lymph nodes. And that hasn't happened yet. That takes some time. So before that happens, the IgM is made, and because there are 5 copies, it can have higher avidity for the antigen. So the individual antibodies may have low affinity, but because there are 5 of them, has higher avidity and can do a good job at clearing the pathogen. But eventually, we can make high affinity antibodies, and those are in the form of IgG, which are a single dimeric antibody molecule. Also on this slide is shown IgA, which you can see takes longer to make, and in the serum is lower titer. IgA is mainly an antibody present at mucosal surfaces. So if you're infected with influenza virus, you will make IgA at your mucosal surface. You will also make serum immunoglobulins, but you'll make IgA at the surface, and that, of course, is important for preventing infection where the virus is entering. So IgA is also made by, of course, B cells, but in this case, the antibody doesn't remain in the circulation. And here's how it gets out. Here's an epithelial cell. It could be respiratory epithelium or gastrointestinal. Here's the apical surface with the villi and the lumen and the basolateral surface below. And here is a plasma cell, a kind of B cell which is adapted to making lots of antibodies. And this B cell has arrived here from the circulation. It's been attracted to these basolateral surfaces. And this particular one is making IgA, which is destined to be secreted. And the way it is secreted is IgA is made as a dimer, polymeric IgA. As you can see, two antibody molecules linked together, which then is secreted from the plasma cell under the basolateral surface of the epithelial sheet. And then the antibody binds a receptor, the polymeric Ig receptor, on the basolateral surface of this cell. The IgA binds it, it's taken up by endocytosis, and it is released at the apical surface. So it's called transcytosis because it goes from one side to the other of the cell. And on the cell surface, the receptor linked to the IgA would be released, and you would still have the receptor bound to the membrane. So there's a protease that cleaves off the receptor. So you have now IgA released, still attached to part of the receptor. And that's called secretory piece. And so you have two IgAs linked to secretory piece. So this is how secretory IgA gets from within us to the cell surfaces. But of course, these plasma cells are producing IgA in our tissue. So it gets into the circulation as well, which you can see from the previous slide. Now, if you happen to have a virus infecting this cell, which is shown here, these yellow markings are viral proteins, it's possible that the IgA and the viral protein can intersect at some point, so that IgA might bind the viral protein and stop infection there. So the IgA need not just work in the mucosal surface, in the mucus actually. It can also work in these respiratory or gut epithelial cells. Now, an important concept here I want you to understand is what is a neutralizing antibody. When a virus infects a cell, and dendritic cells take pieces of it and present it to B cells or T cells, in this case B cells, you can have lots of different antibodies made, but only a subset of them will be neutralizing. And what I mean by that is that it blocks infection. That's what neutralizing means. Neutralizing antibodies. There's a whole set of antibodies made against the virus, but a fraction of them are going to be neutralizing. Many of them will bind the virus particle, but will not block infectivity. These are an essential defense against many viral infections. That's why we vaccinate, to induce the production of not only neutralizing antibodies, but memory B cells of those antibodies that will become activated on a second infection. So they're essential defenses. They neutralize virus particles, and we'll look at neutralization in just a moment. They can neutralize virus in the blood, but not all viruses end up in the blood. They may remain on our mucosal surfaces, respiratory or gastrointestinal surfaces. These antibodies can neutralize virus particles there as well, or in other tissues, need not be in the blood. But it so happens that there's a high concentration of them in the blood, and we measure them often by looking in the blood. So the antibodies in your blood, if the virus happens to spread in the blood, they can neutralize infectivity. Of course, if the virus doesn't enter the blood, they're not going to have any effect there. The IgA mucosal surfaces will block entry of virus, and it's typical that respiratory pathogens or viruses, to prevent infection with them, you need a good IgA response. Influenza virus, rhinoviruses, adenoviruses, for example, respiratory pathogens. If the vaccine you're making, for example, doesn't induce a good secretory response, it won't be effective at preventing infection. Most of the neutralizing antibodies we think of as preventing infection. They're essential defenses. So when you're first infected with a virus, you're eventually going to make antibody, but it will probably be too late to cure the infection. It's not going to prevent it because it's already established itself. If we're in the adaptive response, the virus has already gotten past innate defenses and it's replicating, and it's more likely that cellular responses, CD8 T cells, are going to resolve the infection eventually. But if you have a memory response to the virus in form of memory B cells, the next time you're infected, then those antibodies will be produced very quickly and they can prevent infection. So it's important to remember the difference between your initial infection and preventing a second infection. Antibodies are a good defense for those second infections, and that's why vaccinations work. We do know for some virus infections, though, that antibodies are important for recovery. It is not all of them, but in many cases, if you take away the antibody production of an animal, recovery from infection is prevented. Now, we typically think that recovery, as I said, is mediated by CTL responses, but there are a few examples where antibodies are important too. So here's an example of what neutralization is. This is a plaque assay. It's actually a fake plaque assay. It's circles drawn on a blue background, but it illustrates the point. Here we've got 15 or 10 plaques on a plate of cells. And at the top, what we have done is before adding the virus to the cell monolayer, I assume you remember what plaque assays are, you mix the virus with antibody. And if it's a neutralizing antibody, it's going to prevent plaque formation. And here we have dilutions of antibodies at the top here. We take some serum from an inoculated animal. Or we could take serum from you and ask if you have antibodies to poliovirus. We could make dilutions and mix them with poliovirus. So 1 to 10, very concentrated serum. 1 to 100, 1 to 1,000, 1 to 10,000. So you can see plaque formation is prevented at the higher concentrations, lower dilutions of anti-serum or serum. So the controls are at the bottom. We are not adding any. We're probably adding serum from a unimmunized control animal. So you want to have the same serum proteins added to the virus. You can see there's no neutralization. There's no blocking of plaque formation. Eventually, you dilute out the antibodies from the serum. So at 1 to 1,000, you see we get 2 plaques. So our titer now is being reduced. And at 1 to 10,000, we get as many plaques as with the serum from the non-immunized animal. So we've diluted out all the antibodies. So from this, you can get an approximation of the antibody titer. It's usually the inverse of the dilution where you get 50% neutralization or so. And you will see that reported as when people talk about antibody titers, this is the kind of assay they do. It need not be a plaque assay, but some kind of assay that is preventing infection. Now, antibodies are important for preventing infection. I've said this several times, and that's why vaccines work. But here's an experiment that demonstrates that. We have animals that are being infected with poliovirus. We have given these animals different concentrations of anti-serum against the virus, which has been raised in another animal. So you've inoculated an animal with virus, take out their serum, which you show has neutralizing antibodies in it. And now you're giving those antibodies to a second animal. This is called passive immunization. And we're looking at percent paralysis. So this is poliovirus, which is paralyzing these experimental animals. They're probably monkeys. This is an old experiment, and that's all we had at the time. So our control group of animals, they get serum from a non-immunized animal. They're all paralyzed. And then here we are putting increasing amounts of serum from the immunized animals into these at the time of inoculation with the virus. So at a titer of 3, all the animals get paralyzed. There's no protection. We increase the titer of antibodies. You get about 50% protection, a little more as we go higher. And then at this concentration of serum, all the animals are protected. So basically, we are going from left to right. So we're increasing the amount of antibody given to these animals. And you can see that at a high enough concentration of antibodies, they're protected from infection as measured by paralysis. So that shows you that antibodies can prevent infection. All right, so antibodies can prevent virus infection. There are many, many experiments like that out there. And of course, vaccination is the same approach. We do vaccination that generates antibodies in you and memory cells that can prevent infection. We'll talk more about vaccines later on in its own session. So here is a schema that answers the question, how do neutralizing antibodies work? I've told you that some antibodies against the virus can block infection. Well, how do they do so? And to illustrate that, on the left we have the pathway of entry into a cell of a rhinovirus, which is binding a cell surface receptor. It's being taken up by endocytosis, and then when the pH drops, the RNA comes out of the particle and initiates the reproductive cycle. On the right is shown the different steps where neutralizing antibodies can interfere with infection. So first, the antibodies might bind the virus and block attachment. They might bind to the proteins on the virus surface that are involved in attaching to cell receptors. So if they block those sites, the virus cannot attach. But that's just one mechanism of neutralization. A second one, some antibodies actually do not block attachment, but they block endocytosis. They interfere with the uptake of the particle into the cell. Another set of antibodies can actually block uncoating. So the virus, with the antibodies bound to it, can bind the receptor, get taken up by endocytosis, but it ends up being in the endosome, not being able to have its RNA come out because the antibodies are somehow preventing the conformational changes that are needed for the RNA to get out. So those are three different mechanisms. And number four, some antibodies have been shown to actually be able to neutralize after replication starts. So you infect the cell, and the RNAs in the cytoplasm starting to be translated, and the antibodies are actually taken up separately by endocytosis, and they intersect with the virus at some point in the infected cell, and they block replication. So those secretory IgAs that I showed you before, where they can intersect viral proteins in the cell, that's an example of that. And finally, some antibodies cause the virus particles to aggregate. Big groups or clumps of aggregated virus particles because they're cross-linked. You know, one antibody has two combining sites, so it can bind two virus particles. You can imagine a big lattice of viruses aggregated. They can't get into cells, so infectivity is blocked. So these are the mechanisms by which we think neutralizing antibodies work, how these antibodies block infection. Now, you may intuitively say, oh, they must all block attachment, but it's not the case. Attachment is just one of many mechanisms by which they can work, and I want you to remember that. So I've told you just now that neutralizing antibodies can bind virus particles, okay? There are proteins on the surface of the virus particle, and the antibody is specific for one of those proteins. In fact, technically it's an epitope, a short amino acid sequence, 12 amino acids is recognized by the antibody. So that epitope is on the surface of the particle, the antibody is bound, and that can result in neutralization, as I've shown you, by 5 different mechanisms. If this is an enveloped virus and there is an antibody bound, it can collaborate with complement, the complement set of proteins in serum. Complement will recognize that an antibody is bound, and it will actually lyse the membrane of the virus particle. So this only works for enveloped viruses. So there are 2 ways that antibody binding to virus can block infection. One, by the mechanisms of neutralization, which I just told you, and secondly, by complement-mediated lysis, and it can also be phagocytosed by virtue of the complement proteins being there. Antibodies can also affect the infected cell, as shown at the bottom here, and I implied that in the previous slide, that antibodies can actually get into cells and inhibit viral replication. So here is one way that can happen. So antibodies can bind to viral proteins that are displayed on the cell surface. So as you know, many enveloped viruses, as they replicate in cells, they display viral glycoproteins on the cell surface prior to the budding process occurring. Those proteins can be recognized by neutralizing antibodies and lead to destruction of the cell in a variety of ways. And those are shown here. So you can have lysis of the infected cell by complement-mediated mechanism. So again, by having an antibody bound to a viral protein on this cell, complement can lyse it. You can have inhibition of viral replication. The antibody might get in, or it might just remain bound to the viral glycoprotein and signal into the cell in a way that inhibits virus replication. Sometimes these antibodies bound will block the budding process. They're on glycoproteins that are destined to be in the budding virus, and the antibodies block that process. And in some cases, these antibodies will inhibit cell-to-cell transmission. When the virus particles bud off, these antibodies can block them from getting into other cells. So the point that I want you to take away from this is that antibodies can block virus infection of cells, or they can take an infected cell, a virus-infected cell, and also inhibit the production of viruses in those. And that's why they are protective. That's why these antibodies are so powerful. Here's a picture, one of my favorite pictures of poliovirus, made by Jason Roberts, who is a virologist down in Melbourne, Australia. And he made this for the cover of our textbook for us. So this is poliovirus particle, and you can see the red and the green and the blue proteins, 3 of the capsid proteins making up the icosahedral particle. And bound to it is a single monoclonal antibody. That means it recognizes a single epitope on the viral capsid. And so these are the monoclonal antibodies in purple. And what you can see is that there are 5 around each 5-fold axis of symmetry, exactly as you would predict. For any epitope on the capsid, there would be 5 of them around the 5-fold axis. And since there are 12 5-fold axes, you have 12 clusters of 5 antibodies bound. So it's perfectly aligned with icosahedral symmetry. Now, this happens to be an epitope that is at the 5-fold axis of symmetry, but there could be epitopes at the 6-fold axis or anywhere else in the virus particle, and it would be similarly repeated. But the point here is that this is a neutralizing antibody that recognizes a short amino acid sequence on the surface of the particle, and that's how it blocks infection. The mechanism may be any one of those that I mentioned to you, but it certainly is neutralizing. Viruses can escape neutralization in a number of ways. And there are 2 on this slide that I want you to understand. And you may say, well, if the host is making antibodies against the viral protein, how can it escape that? It's called antigenic variation. And one embodiment is shown on the left, rhinovirus. So you get infected with a rhinovirus, your body makes antibodies, neutralizing antibodies against it that block infection. And then next winter, you get infected and you get a cold. You get no protection from your antibodies. Why not? Because there are over 150 different kinds of rhinoviruses. So you do the math, you could have 2 every year and never see the same one for your first 75 years of your life, I guess. So the strategy there is that the virus makes lots of antigenic variants, which have different sequences in the capsid proteins so that antibodies against one will not bind the other virus, okay? So 150 different rhinoviruses, you make antibodies against one, it neutralizes that one beautifully. But next year you get a different one, and these viruses circulate everywhere. And here in New York, we could probably find 20 different types of rhinoviruses and 20 different types somewhere else, so you're not likely to get the same one. So that's one strategy. And the other is shown on the right by influenza. This is a virus we'll come back to several times. It undergoes antigenic variation of its glycoprotein. So there aren't 150 different types of influenza viruses, but there are a few types that vary tremendously in their protein sequences. So what we're showing here is the hemagglutinin of influenza virus. And remember, that's a viral glycoprotein. It's in the viral envelope, and the cell receptor attaches on the top there. And these colors all represent epitopes that bind individual monoclonal antibodies. You can see there are a bunch of epitopes at the top and on the side as well. And when you make neutralizing antibodies against this virus, they bind these particular epitopes to block infectivity. The problem is from year to year, the virus can change these epitopes. One amino acid change in any of these epitopes is enough to evade antibody binding. And so this is what the virus does from year to year. It undergoes antigenic drift. As it multiplies in a population that has antibodies against it, variants are selected that escape neutralization. And this is a dynamic process that happens every season from year to year. And that's why it's hard to make a vaccine against this virus, because the next year it's going to be 1 or 2 amino acids different, and it won't be effectively neutralized by the vaccine. So those are 2 strategies of escape from neutralizing antibodies. There's a third one here in this article, poliovirus escapes antibodies. And this is actually a story about a natural escape where poliovirus variants arose in a country that wasn't sufficiently immunizing against the virus. So you had low levels of population immunity, and variants were selected that had, again, single amino acid changes that could overcome neutralization. And I present that because polio is considered to be one of these viruses that doesn't undergo much antigenic variation, but there are specific examples where that can happen. For poliovirus, we have used the same vaccine for over 50 years. There is not sufficient antigenic variation to not use it. But in this one example, in one country, a variant did arise because they weren't using the vaccine properly. Next question. Which statement about antiviral antibodies is incorrect? Incorrect. They are important for protection against viral infection. They only neutralize virus infectivity. They may block virus attachment to cells. They can be found at mucosal surfaces. IgM is the first to appear, then IgG. So most of you got the right answer. They only neutralize virus infectivity. They do that, but they also do other things as well. They help lyse the infected cell and so forth. Everything else is correct about them. So the other half of the adaptive response is cell-mediated immunity, where a cytotoxic T cell will lyse the infected cell. So this doesn't operate at the level of virus particles, but of infected cells. And this, as I said earlier, is important for clearing most viral infections, although there are a few exceptions where antibodies are important. What happens is the cytotoxic T lymphocyte, this is a CD8 positive T cell. It's been activated by interaction with a dendritic cell. It's expanded, so now that there are many of them that recognize the original peptide presented by the DC, and now they look at an infected cell, and if they find that peptide displayed in MHC1 now, not 2, but MHC1, they will kill that cell. So it involves formation of a synapse between the CTL and the infected cell. It causes lysis. And of course, viruses antagonize this as well. But the key here is that this has to be an infected self, which means it has to have MHC1 on the surface, and it has to present that antigen to which the CD8 has been selected to be specific by the interaction with DCs. So let's see how this works. So this involves the endogenous antigen presentation pathway, including or utilizing MHC type 1 molecules. The other pathway we looked at, MHC2, is restricted to antigen presenting cells. This MHC1 is found on all cells, and that's how the peptides of viruses are presented. So it's, again, a plasma membrane protein, and here we have an infected cell on the bottom, and it's being queried by a CTL at the top. So the T cell receptor is looking for the peptide embedded in MHC1. It's called endogenous because these are virus-infected cells. They don't take up exogenous protein as the DCs do, but they're infected. The virus is infecting this cell. Viral proteins are being made. And what happens in these cells is that some of the proteins are degraded by the proteasome, a very large proteolytic machine that's present in cells. And the peptides that are made, those should be those familiar orange boxes. They are transported into the endoplasmic reticulum via a transporter. TAP. TAP is called transporter associated with antigen processing. It's actually a channel that moves the peptides in actively. And in the ER, the MHC1 molecule has been made and assembled, and the peptide is loaded into the MHC1, and that complex then makes its way to the cell surface, where it can be queried by the CTL. And if it's a foreign peptide, the CTL will lyse that cell, but it has to be presented in the context of class 1. So that's the endogenous pathway. These virus-infected cells will display these peptides on the surface, and they will be killed, which you may think, wow, that's pretty dramatic or drastic, but that will limit the virus infection. You kill off, less than 1% of your respiratory epithelium, but if that prevents the infection, that seems to be a beneficial outcome. Now, viruses interfere with multiple steps in this class 1 presentation pathway. And again, these tend to be herpes viruses that cause long-term persistent infections. They're not cleared because of these evasive mechanisms. So for example, herpes viruses can interfere with the transcription of MHC1 and other proteins that are needed for its display on the cell surface. They can result in the retention of MHC1 in the ER, so it never makes it to the cell surface. They can inhibit the transporter, so the peptides never get loaded. What's another one? There's another one where some viruses encode proteins that downregulate MHC1 from the cell surface. They induce its endocytosis. So all of this results in no MHC1 on the surface or MHC1 without a peptide. So even though the cell is infected, it's not detected by the CTL. So it's an amazing strategy. These viral proteins can do all these things to interfere with class 1 dependent display of peptides. So they don't get killed because the CTL doesn't recognize it as infected. Here are some of the viral proteins that counter MHC1. As I said earlier, there are many more countering mechanisms than we saw for MHC2. And you don't need to know the proteins. I just want you to understand the targets. For example, MHC1 synthesis can be interfered with, in this case by a HIV protein, synthesis of the transporter for peptides into the ER, or its function can be inhibited by different viral proteins. MHC1 transport to the cell surface can be blocked. It can be retained in the ER. It can, in this case, dislocate the MHC1 to the cytoplasm so it doesn't get shuttled up to the cell surface. And as I said, you can increase MHC1 endocytosis. So there are different viral proteins here that have all these various activities. And this should tell you, when there's so many antagonists, this should tell you that this is a really important pathway, because if the CTLs can sense that a cell is infected, it will be destroyed and the infection will be stopped. So this is a way of having the infected cells hide. And as I said, that's why these herpes virus infections are difficult to clear. How does the lysis occur? Two mechanisms. The CTL makes a very tight junction with the infected cell and makes two kinds of pore. It makes a lytic pore in the membrane, which is shown in panel A, and that causes the contents of the cell to leak out, and the cell will eventually shrivel up and die. Or it can make a different kind of pore called a perforin pore, and what it does is it sets up this pore in the target cell and puts in a protein called granzyme B, the little red dot, goes through the pore, and that induces apoptosis, programmed cell death. These are two ways that the CTLs can kill these infected cells by release of cytoplasmic content or by apoptosis. When an animal is infected with a virus, you can measure CTL production, cytotoxic T lymphocytes, CD8 positive T cells. Here on the top is virus titer. So this is an animal experimentally infected with a virus. And we're looking at virus titer here in the spleen in this particular animal. You can see very rapid peaking of virus by day 5 or 6, and then a rapid decline. And at the bottom is the induction of cytotoxic T lymphocytes. And in green is the total population of cytotoxic T lymphocytes. You see they peak at about the time when the virus titer is going down. And the purple are epitope-specific ones. So these are partially memory cells that remain for a long time. They are specific for the epitope, and they persist. So here's an experiment that shows you the role of antibody versus cellular immunity in protecting animals against monkeypox virus infection. And so what we have done here at day 0 for all the groups, we vaccinate the animals with an inactivated virus, a monkeypox virus. On this, then we do some kind of immune manipulation, either nothing in the control group or something else in the others. And then We measure at day 22 neutralizing antibody in the serum. We do a neutralization test. Then we infect with monkeypox virus, infectious virus at day 28, and then we look at fatality. So when we vaccinate these animals and do nothing to manipulate their immune system, they make a good neutralizing antibody response, and there's no fatalities. So they're protected by the antibodies that have been induced. If we deplete these animals of B cells after immunization, you see they don't make any neutralizing antibody. When you infect them, 3 out of 4 die. So this tells you that the antibodies are important for protection. If we deplete CD8 cells, you still get a good antibody response, and you would expect that because CD8s don't play a role in that. If you depleted CD4s, you might see a difference, and these animals are all protected. So in this case, antibodies are important for protection. You could do similar experiments in another virus which would show that CD8s would be important. Now, it turns out that, as I said earlier, some viruses, the CTL response is more important than the antibody response in protection and recovery. So the question arises, how do we know which response to make, whether we make an antibody or a CTL response? And that begins in the lymph node where the dendritic cells, the sentinels, encounter B and T cells of the virus. And the way that is determined is by the helper T cells. Remember, the dendritic cell comes into the lymph node. It makes contacts with a variety of T and B cells. Among those are T helper cells, the CD4 positive T cells. And the information that is exchanged between the DC and the T helper and vice versa will determine what the T helper cell differentiates to become, a Th1 or a Th2 cell. And remember, the Th2 response, so a helper cell making mostly Th2 cytokines, causes the production of antibody-producing B cells. So the Th2 response favors antibody. In contrast, the Th1 response favors the production of cytotoxic T lymphocytes. And all this is controlled by the kinds of cytokines that are produced and the peptides that are presented by the dendritic cells. And all happens initially in the lymph node, and typically the cytokines produced that lead to Th2 responses inhibit the Th1 response, and vice versa. So here on the right is an example of an antigen-presenting cell that's sensed a certain virus or viral protein. It's making IL-12, which happens to be one of the cytokines that favors the Th1 response. It favors the production of CTLs. So for this virus, it has evolved to be one that requires CTLs for protection, and that's how the response is mediated by the production of very specific cytokines. You can see no Th2 response because apparently they are not needed for protection. And it can go the other way as well. If the virus needs to be cleared by antibodies, cytokines will be produced that favor a Th2 response. So the whole thing is determined in the lymph node, whether you're going to make antibodies or cytotoxic T cells. And most of the time, of course, it's not black and white. There's a mixture of both. But these cytokines that are initially produced are determinants of that decision. Our last question for today. For some infections, CTLs are more important for protection than antibody. How is the CTL antibody balance determined? By toll-like receptors, by intrinsic defenses, by autophagy of infected cells, by the mix of peptides and cytokines presented by DCs. It depends on whether the capsid is icosahedral or helical. Most of you got the right answer by the mix of peptides and cytokines presented by DCs. Now, these responses that we've talked about, both B and T cells, provide memory because you go through a lot to make specific antibodies and CTLs. Why waste it? So if you were infected again, you're going to get a rapid response, specific response. As I showed you earlier, it's not going to be delayed, it's going to be more robust. And this is a characteristic of adaptive responses, that they have memory, and it's not something that is a feature of innate responses. And this memory, of course, is why we vaccinate. We provide an encounter with the virus without pathogenic consequences that provides memory, so that if you ever encounter the virus, you will make a very robust B or T cell response and be protected. Memory was first discovered by an epidemiological study with measles virus. And this was a study done in these very remote Faroe Islands, just north of the UK. And there was an outbreak of measles on these islands in 1781. Back then, they could only be reached by boats, right? And so there was a good maritime trade with the island, which would bring measles in periodically. And there was an outbreak in 1781. After that time, actually, a lot of the population was killed, and the maritime trade decreased. So for the next 65 years, there was no measles on the island. And then as the population restored, the trade began again, ships started to come back. There's another outbreak in 1846. Again, someone brought the virus in on a ship, but none of those who had survived the earlier epidemic were infected. They had memory. And this was first studied by an epidemiologist who said, he didn't know what the virus was. There's nothing known about viruses at this time, but they knew the concept of infection and being protected. It was known from smallpox, and they said there must be some kind of memory, and this showed that it lasts a long time, and it doesn't require re-exposure to the virus to maintain this memory. And memory is demonstrated in this graph. You have your first infection, and you measure either antibodies or T cells on the y-axis. You have your initial response after 7 to 14 days. It then declines. And antibodies in your serum or T cells are very low, but they're there. But more importantly, there's memory, so that if you encounter the pathogen again, it can be years later, you will have a very rapid and more substantial response. And that is enough to interfere with infection, whereas the initial response was too late. So that is immune memory. It's maintained by cells that are related to those that were initially selected by the dendritic cells in the lymph node. So we have what we call memory B cells that persist in various parts of the body. So remember, the initial DC selected out a B cell that then went on and did recombination and somatic mutation till it made a very high affinity antibody. So that is stored, so you don't have to go through all of that process again. And when the virus comes back, those cells are selected and they amplify very quickly. They don't have to undergo all of these processes to make a high affinity antibody. We also know that there are some plasma cells that live for a long time, and these, of course, secrete lots of antibody, and they can respond as well. And there are also memory T cells. Same idea, that you have selected out certain T cells that are specific for viral epitopes. There's no need to go through the whole selection process again. All you need to do is amplify them, and that is a memory T cell. Now, all of these activities happen in different parts of our bodies. We've mentioned the blood, but our skin has a very specific immune system. Here, for example, is our epidermis and dermis, and there are a variety of cells in the epidermis that are patrolling and looking for infections. Here, for example, is a blood vessel. And we have keratinocytes, which have class 1 molecules on them. They're able to make a variety of cytokines and respond to infection. There are dendritic cells that are specific for the skin. They're called Langerhans cells, and they can pick up foreign peptides. They can actually interact with T cells that are resident in the skin. So these dendritic cells will eventually go to the lymph node, as we talked about before, but to make a quicker response, there are resident T cells of two kinds, Th1 and 2, in the skin that can respond, so you get a very immediate local production. So that's the skin. The same goes on in the gut. We have a mucosal immune system in the gut. Here is the gut lumen at the top and our lining of epithelial cells. And beneath the epithelium, we have a variety of cells, including antigen-presenting cells, the dendritic cells that can present antigens. We do have resident T cells in this subepithelial space, but of course there are blood vessels and lymph vessels, so that dendritic cells can also reach local lymph nodes to instruct B and T cells there as well. But again, just like the skin, you have T cells present, so you can initially make some T cells, or you can even make antibodies before going to the lymphatic vessel. And we also mentioned a while ago these M cells, which are interspersed among the epithelium. These are very good at transcytosing antigen from the gut lumen. That antigen can be sampled by dendritic cells to see if it's foreign or not, and it would include viruses. Of course, this is called the gut-associated lymphoid tissue or mucosa-associated lymphoid tissue. So I want to give you a last overview of this picture and try and summarize it by saying that these two systems, the innate and the adaptive system, they're integrated by inflammation. And that's kind of summarized on this slide where we have at the top a lymph node and a lymph capillary, and at the bottom a blood cell. And in the middle we have some viral antigen, in our case, as a consequence of an infection of a cell. It's being sampled by dendritic cells, of course, which are producing cytokines that contribute to inflammation. The dendritic cells go to the lymph node, produce T and B cells, which will come back to the infected area. At the same time, complement is also acting. The detection of patterns on molecules by complement can lead to the production of a variety of proteins that further cause inflammation. And a variety of immune cells can produce substances that will dilate the capillaries. They make spaces between the endothelial cells so that cells can go in and get out, and some of the cytokines that are produced can help prepare the blood vessels for cells going in and out as well. So it's a very interesting interaction between two specific arms of the innate and the adaptive responses, and inflammation is what links them, the production of cytokines that tell cells where to go and how to deal with the antigens. So now we have a kind of overview of defenses against infection. What I want to do next time is to put this together. We needed to do this because, as you will see, a lot of the pathology caused by viruses is immunopathological. So next time we can talk about the different mechanisms by which viruses cause disease.